All right, so I'm doing my project, my capstone on vegetation change on Santa Rosa, and that's the whole island as a whole. So my hypothesis, or I have a couple, but the main one is the vegetation will change, the change will be greater in areas that have steeper slopes and that the accessibility is different for cattle. And that was based on 3 groupings, 0 to 10% being very accessible, 10 to 30 being moderately, and 30 to 90 being pretty much no accessibility at all. And then some other things that will affect the vegetation change will obviously be aspect because of a prevailing wind, because of solar radiation, because of water, and stuff like that. So I'm going to do it by, it's an ongoing project, it's a timeline project, so right now I just finished a 2015 vegetation map, and that's what this bottom one is. And the top one is the original Landsat photo that came from USGS, and that's 7 different bands, and that's how I was able to build these vegetation categories. So there's 6 different time series from 91 to 93 to 98 to 2010 to 2015. And my methods, the map on the right, it shows the slope categories. So the green is the 0 to 10 slope, and then the yellow is the 10 to 30, so the moderately accessible, and the red is the 30 to 90, so not accessible. And this map is aspect of the island, so just the aspect of different hills and shading that is occurring on the island. And what I have so far, I extracted the vegetation from 1993 to 2015, and I compared the difference in vegetation. So I was able to do that I overlaid my 3 categories of slope onto the map, and I extracted the pixels. So by extracting the pixels from 93 to 2015, I can see the difference in how many pixels are in each map. And from that, I can convert it to kilometers squared. So this graph right here shows that vegetation is increasing for sand and for scrub and coastal bluff and things like that, and we're actually seeing a decrease in bare ground and the largest decrease in foothill valley grasses. And thank you.
All right, hi, my name is Erica Tirado, and my capstone is on environmental attitudes of underrepresented students in Oxnard, California. So my hypothesis, I hypothesize that students will have a greater connection to nature after participating in this unit or program. That's called Crossing the Channel. So this is my method. So I'm using the Crossing the Channel program, and it has 4 units. The first unit was on biodiversity, second was watershed, and then the third is long-term monitoring, and this last one is environmental stewards. So this program also gives the students field trips, which gives them experience out in the field. And so I gave them a survey at the start of this third unit, and I'm giving them a second one after this unit is over. And for my results, I hope that this is the beginning, this blue color, and their IQs were very low. Most of them were like, well, I don't want to go out and do science and stuff like that. But afterwards, they were really excited about it, and that really showed in the post-survey. They were really excited about it, and they were telling me that they really want to actually go out there and do this stuff now. And next time they see a sand crab, because that's what we were monitoring for in Carpinteria, they actually want to count them now and do that sort of stuff.
Hello, everybody. So I'm working on solar radiation and how it can be measured on the ground. This photo is pretty interesting. It's an experiment where they're actually trying to trace photons. So there's a machine that makes photons like the sun would make, and so this is kind of trying to color trace them and see what direction they go and stuff. So it's kind of neat. And so the topic here is direct solar radiation and topography. How does the level of measured direct solar radiation vary with changing ground elevation in Ventura County. So that's kind of the question to be answered, right? So yeah, we all know about that. So the hypothesis, or first I want to say this little quote from Ruiz Arias et al. In their paper. Solar radiation is a major forcing function of physical and biologic processes on our planet. Knowledge of the spatial variability of radiation components is crucial in order to understand these processes. So I think it's one of those things that kind of flies under the radar. You know, people know about it. People know, okay, sunlight is important for photosynthesis, et cetera, hydrology. But to further understand these processes requires a little more work that I think hasn't really been done up until now, until some more recent studies. And so this particular project is concerned with how solar radiation levels vary with elevation changes. So the hypothesis, my hypothesis, measured daily direct solar radiation levels would be greater as elevation of the sampling site increases. So you go up to higher elevations, you get higher solar radiation levels. So the weather station that's on the roof of the building right above us is kind of a control unit, right? So that's a fixed unit that doesn't move. It's measuring all the time, and so that's a good baseline of knowing kind of a ground level radiation level for a control. This is the TES solar power meter. So it has a little pyranometer on the top here, and it's basically the same thing that the weather station has. And so I can take this wherever I need to go. And so I can measure the solar radiation in kilowatts per meter squared. So there's some locations and their elevations, and some features and what they're all about. This is a map from the National Renewable Energy Laboratory, which is a government agency that is looking at solar power, that kind of thing. So we're going to see how we can relate the data I collect with what they have collected. So here's some results, a sample graph just showing the course of sunlight throughout the day and what kind of radiation levels you get. About 9.9 kilowatt hours per meter squared is kind of on average as a high, especially for this area. So we're going to see how that varies. And I wanted you to meet Kai. I call him the anti-capstone. This is my son. If it was up to him, I wouldn't be here right now because he would.
Boom.
I'd be careful about spending more time on your kid and less time on what you're actually trying to present. You know what I mean?
What are you saying?
You gave an introduction to your son, but you didn't get a chance to show us what you wanted to show us.
I knew someone was gonna give me crap for that.
No. It's not crap for showing your son, man. It's just we wanted to see the data, right? Because that's what we wanted to hit. Yeah.
Anyway.
Hi, I'm Vanessa. I'm studying coral reef health in the Cook Islands, and so basically what I'm studying is when I first started off with answering the question of does the proximity to human inhabitants influence the health of the reef? And after a few manipulations of data, it turns out that that was not the whole problem. So it grew to now focusing on creating a health index of the reefs in Aitutaki, both comparable to global health indexes as well as specific to the island. And then with that, I am going to create management solutions for the problems that they're facing there and sending it to the government. So this is the methods. This is what we did. So we laid out transects at 12 sites in Aitutaki. And then we looked at substrate under a point intercept form, so every 20 centimeters. We took the substrate, we measured and counted all of the invertebrates. We identified fish, and then we also took the reef height to look at the structure. So this is kind of what I'm finding. So sea cucumbers are filter feeders, and so they're an indicator of excess nutrients in the water. So the Cook Islands still uses DDT and other agricultural pesticides, and so you can see, so the main little island there in the middle is where everybody lives, and so there's a higher abundance of sea cucumbers around there, which means that some of the things that they're doing on the mainland could be impacting the water. And this is the first analysis for a global comparison of live coral cover in Aitutaki. So these are designated categories for like global live coral, and as you can see, there's only one reef in Aitutaki that actually is healthy, and it's the only protected reef in the area. So they're kind of, now the question comes, is protection the only way to save the reefs? And that's what I'm going to determine. That's it.
Hi, my name is Jay Woolsey, and I'm looking for expansion in the Quercus tomatella groves on Black Mountain on Santa Rosa Island. So my hypothesis is that since the removal of non-native grazers, we're going to be seeing expansion in these grove areas because the grazers aren't coming in and eating all the saplings. So issues with this project is projections from old maps using old technological systems have provided us poor data, and we're seeing problems with that, as you can see in this lovely map right here. So methods. I went around first to the groves and dropped a GPS outlier or a GPS point on each tree, creating polygons. Then I went to the hug in the trees. So then I went to all the outliers and I took DBH, height class, GPS points, and if they were reproductive or not. Then from that I went into ArcGIS and I looked at kernel density to show how dense these groves were and to look at what the future of these groves could be. And I looked at polygon area to see if the groves in fact had changed in size. Then I ran ANOVA's frequency and regression test to see what was going on with the outliers. Are they old outliers? Are they young outliers? What's going on there? So then I went to my results. Groves had expanded by 20%. There are over 1,600 outliers in the groves. I got an ANOVA p-value of 0.001. And that helped me see that there is a strong correlation between DBH and height class. The higher the DBH, the higher the height class. And then I ran some just general statistics on what DBH was, on what the DBH was. Apparently some things got messed up in between PowerPoint and Google Docs, but that's all good. So I got a mean of about 5, median of 3, standard deviation of 8.6. The minimum DBH was 0.6, and the maximum 143.9, the big boy. All right, wait, sorry, that was supposed to, oh, no, okay. That's the last slide.
Hello everybody, my name is John Slagboom, and my capstone project is to establish a baseline of emissions from 2-cycle and 4-cycle gas-powered landscape equipment and on Channel Islands. So let's go into a little bit of a background of why that's important. So in 1995, the EPA said, wait, hold on a second, we're measuring the emissions from vehicles, but what about everything that is not on the road, like not semi-trucks, not standard consumer cars, what about like all the forklifts and so on and so forth. So in 2011, they realized that for hand-powered equipment, so like a weed eater, a lawnmower, a string trimmer, they don't have emissions data for that. And in 2011, they started their first study that found that approximately 27 million tons of emissions, criteria pollutants, are emitted annually. And within that category, 2-cycle engines are responsible for a majority of those emissions. So they're like, whoa, hold up, we're generating a lot of emissions, we got to start narrowing down where these are coming from. So enter my research. We have a lot of modeled projections of what those emissions look like at regional, state, and county levels. That's just by taking the horsepower of equipment, how often they're used, and so on and so forth, extrapolating that information. But we don't have official documentation or any really rough idea of what's going on at the community level, at the facility level, at the household level. So that's what I'm seeking to do with CSU Channel Islands, is take information based off the emissions of actual equipment here on campus, extrapolating it to the population and the equipment of the campus, and then saying, okay, this is a baseline. So let's look at some of the results. Actually, let's talk about the methods first. So I took a census of the equipment used by the landscaping crews. I randomly selected representative individuals from each different type of equipment category, and over a period of 4 weeks, we took the equipment, measured each individual twice during the day, one time in the morning and one time in the evening, during midday, and I'll get into the reason later, and then we applied that emission data back to the overall population. The reason why we took the emissions data in the morning and in the afternoon or midday is because we wanted to emulate what the temperature variation in winter and summer would look like. So by taking emission measurements in the morning before the sun heated up, and in the afternoon, we would get those variations. So we have some overall data that shows that leaf blowers are by far the most polluting, largest generators of emissions, closely followed by string trimmers and everything else. And those are the preliminary results, and I'm looking forward to doing further analysis to break down what that looks like over month by month. So any questions?
Hi everyone, my name is Danielle Martinez and I'm studying the prevalence of invasive plant species on Santa Rosa Island in correlation to park visitors. So pretty much the reason I'm doing this research is to kind of see like how invasive plants are getting spread around the island, since it is a closed system as opposed to like somewhere on the mainland. My hypothesis is that invasive plants will be most prevalent near trails leading to the campground and bunkhouse, as well as in the immediate area surrounding those two. So my methods, I used a GPS device called the Garmin Oregon 650T. I studied 3 points of entry, which include the pier, the campground, and the airstrip. I used GPS locations to measure to mark the locations of the populations of the species I was looking at. I marked what the species was, the density class, so how big the infestation was, the percent canopy coverage relative to the natives, to see like if the spread was, really impactful. And then also, checked out the surrounding vegetation area. So results, pretty much I buffered the points so you could see kind of how big the spread was if it had a big percent spread. Ice plant and blue gum were the most prevalent for this point of entry at least. The percent coverage was roughly 7,000 meters squared out of a 20,000 meters squared area that I sampled for this one. So it's about 35% infested at this point. 85% of the infestations were found within 5 meters from bunkhouses or trails leading to the campground. So it kind of does show that, you are seeing it towards those areas for sure. So yeah, that's pretty much it.